Jade and we're here at the Jock Marshall Reserve. I'm manager of the grounds and facilities here. This reserve has been protected by Monash University since 1961 for research and teaching. Behind me is a state-of-the-art environmental classroom that was built around 2015, and we're going to take a tour. So here we have the Jock Marshall classroom, and this is Agni.
Hi, I'm Agnita and I'm the Teaching Technical Coordinator working at School of Biological Sciences. So my background is conservation biology, but now since I work here, I take care of undergrad units and I set up the lab. So this is where the students come in for their laboratory classes, and this lab holds about 112 students. And let's have a tour. For our undergrad units, we take samples from the reserve, so it's on-site samples, and we use them here to study more about ecology, biodiversity, and for all other units. For units which is related to plant studies, we sow the seeds in the greenhouse, which is just near to the JMR reserve, and we bring it in here for the experiments related to, we study about rice, corn, wheat, regarding what the unit teaches. We start from the sowing the seeds, and students get the opportunity to have a hands-on experience with from growing till finishing the experiment in their undergrad studies.
When you do get a chance to come down here, please come look at the windows. They're amazing, and it does feel like you're looking right into the soul of the reserve. So now that we've had a look at the lab, let's go look at the grounds. So the reserve didn't always look like this. In the early 1800s or mid 1800s, it was actually used for farming and mining, and in early 1900s it was then taken over for an epileptic facility, and in 1960s the university took it over, and that's when this was designated. Over the years there's been a lot of different planting styles taking place, all native, but they did used to house macropods here, and so a lot of the plants were used for that, but now macropods are gone. A lot of regeneration has happened, and this is a result. This building here is what we call the environmental laboratories. This is where a lot of researchers come to do experiments in controlled temperature rooms. There's fish, algae at the moment in here, and then just over here in summer, they're not here now because it's winter. In summer you'll see a lot of tubs set up. They're called mesocosms, and they grow up tadpoles into frogs and then take them back to the laboratory. Walk through the reserve, you will see a lot of these statues around. There's a few different types. These are actually animals taken from museum specimens and printed out on a 3D printer. Particular one is blue tongues. Also through the reserve you'll see these signs up. These are transect lines and are used for education and sometimes research. There are pits and traps located at every single one, and where people use them to look at skink specimens and any other bugs that might have fallen in. And there's a few different types of transect lines as well running through the reserve. Domestic shed. So we have a lot of our researchers go out on field trips, and the bones that they collect are cleaned up by our domestic bugs in the shed. Here's one of our camera traps. So if you've walked past, say cheese, because you'll be uploaded onto the computer. We've seen foxes, rats, and students in pyjamas walk past those cameras at times, so they're always interesting to look at. We also have PTZ cameras running 24/7 that take footage of the reserve, and that's actually uploaded live onto the Jock Marshall Reserve website, so you can take a look at that at any time. well signposted, and as you can see from the sign here, we're heading to Farrington Pier. So this is the start of the wetlands that's heading towards the lake. I will take you up shortly to where the wetland begins, where it actually flows all the way down, filters through biofiltration and into the lake, where it's hopefully nice and clean by then. And this is Farrington Pier and the lake. Just a bit of history. It's called Farrington Pier is named after Graham Farrington, and he actually was a curator of the reserve for 40 years. So far the longest curator of the reserve, and he did a lot of important work in that time. So they've dedicated this pier to him, and this is where all the sampling for microinvertebrates takes place. So this is the lake. Very proud of the lake at the moment. 12 months ago it had almost completely dried up because the infrastructure that fed the lake failed and no stormwater was getting to the lake, and now look at it, it's amazing. Oh, and we have a duck too. We have lots of wildlife come visit us, and we have nearly 40 turtles in the lake, so do look out for those. The reserve also has a really healthy population of Perrin's frogs, and if we're really lucky, they'd love to live in the weather station box, so we'll go and have a look see this. Not today, but you can see they've left their mark in there. So one thing that we've tried really hard to do with the lake is not introduce any fish, because obviously they'll eat all the microinvertebrates. So in saying that, we have a really rich microinvertebrate community in our lake because of that, and they often live right down the bottom. So when we do water collections, we always get a little bit of water from the lake, scoop right down into the reeds and mud, scoop up, and then we wash that out into the bucket and see what we've caught. Also, we're very lucky to have a water sampling station and a weather station on the pier here. So the water's actually pumped from the lake, and you can see the buoys in the lake that's actually holding the sampling lines. They run through, the water's pumped into here, and it's actually, samples are taken and the readings are also loaded onto the website. The pump's not running at the moment, and you can look at things like pH, salinity, turbidity and those sorts of things live, and also the weather station also on the website. Something else to look out for when you're down here is we have a really large, I'm suspecting male kookaburra, and he will follow you around the reserve. So look up when you're down here and see if you can spot him. So over here we have fern gully under the walkway, and we also have what we call a frog bog, and we do have a lot of tadpoles in here. Though as you see, it's quite orange. That's iron leaching from the ground. It's not going to harm the frogs at all. They live quite well in there, but you probably can't see them. Right now we're standing in the middle of the biofiltration route. Up the very top there, stormwater is collected from halls of residence in the car park. It flows through all of this plant material and all the way down to the lake. By the time it gets down to the lake, a lot of the metals and nasty phosphates and things like that are being filtered out, and it makes the water in the lake a lot cleaner.
So these are the samples that we got from the JMR lake. So students get the opportunity to get the samples by themselves and then bring it to the lab, and they have a look at how many tiny invertebrates are in the JMR lab. We also compare the sample with the other lake which is in front of the JMR. So those are the ones which has fish in them, and these ones don't have fish. So students get to see all the tiny bugs through the microscope, and they compare the count and the different specimens in different types of lakes. So from today's sample, we were lucky to see some boatmen, some bloodworm, shrimps, etc. We are at the greenhouse facilities, and this facility is mainly for research students, but for undergrad students who are studying related to plants, for example, food changes in the world or global changes. For those units, we grow plants for the students. So this is wheat. So we have several types of wheat, rice, canola, corn, etc. We will be looking at the growth rate, the leaf structure, leaf length, and leaf count in different plants. And after they grow to a certain height, we fertilize them. So half of them will be fertilized and half of them won't be fertilized. So they have a variety to work with, and students do an online version. So we record everything that we do, and students can work with the data, and they'll do a report on it.
Hi, my name's Sam. I'm in my 4th year here at Monash. I'm currently doing my honours in zoology and ecology. Over the past 3 years of my course, I've spent many hours in the JMR, taking the little dinghy out on the lake or putting waders on and finding cool stuff. Lots of environmentally sort of orientated units you can study in the JMR, from ecology and biodiversity to zoology. So my project on the brown tree frogs involves looking at how geographic variation influences growth and development. So we've taken populations from here in Victoria and some in Tasmania, and we'll see how the temperature differences between the two states influence how quickly they grow and develop.